Thank you for joining us today. Dr. Rizzo, would you like to begin?
Sure. Happy summer, everybody. Welcome to today's Congressional Brain Initiative virtual briefing. We're thrilled to have over 170 people signed up. I know people are still joining. I'm Matt Rizzo. I'm the chair of the University of Nebraska Medical Center Department of Neurological Sciences, and also the chair of the American Brain Coalition, the ABC. The ABC is here together today in a truly collaborative effort with the American College of Neuropsychopharmacology, the Allen Institute, the International Neuroethics Society, the Kavli Foundation, the Simons Foundation, and the Society for Neuroscience, partnering today with the Congressional Neuroscience Caucus, the CNC, to get an exciting update on the world-class brain initiative advances and exciting new developments on the horizon. So the caucus, which is co-chaired by Congresspersons Earl Blumenauer and Kathy McMorris Rodgers, was established over a decade ago in 2010 to build awareness of the key role that brain research plays in a number of ways in understanding ourselves, society, communicating the benefits and progress in this research, and promoting changes in federal policies to support neuroscience research. Recently, Representatives Blumenauer and McMorris Rodgers led their colleagues in a letter to the Appropriations Committee requesting robust funding for the Brain Initiative in the coming fiscal year. And we're really grateful for this. Congressman Blumenauer represents the 3rd Congressional District in Oregon. He's a lifelong resident of Portland, and he was first elected to the House in the mid-90s. On the Ways and Means Committee, he's tirelessly promoted healthcare access and environmental reform, in addition to being a great champion for patients suffering from neurological and psychiatric disorders, and for researchers developing cures, much needed cures for these disorders. So Congressman Blumenauer, please take it away.
Okay. Thank you. Thank you, Doctor, for the kind introduction. Thank you for your leadership. We deeply appreciate the opportunity to share some thoughts and observations going forward. Our Neuroscience Caucus was founded to promote congressional investment in neuroscience research. This is the area, as you know, better than anybody, touches virtually every single American family. It's an area that has stubbornly resisted breakthrough cures, even though we've seen a great deal of progress with the Brain Initiative. We're committed to having the same dimension of improvement as we've seen with cardiovascular and cancer research. Research is the hallmark. Research has been somewhat redirected as we've been dealing with the pandemic. It's understandable. We've seen some significant breakthroughs in terms of dealing with vaccines, but we don't want that to be at the expense of other areas of neuroscience research. This is an area where we can't just start and stop. Frankly, we've been coasting on some of the significant investments that happened a decade ago as part of the recovery package dealing with the last economic collapse, creating lots of family wage jobs, lots of progress, keeping a generation of researchers in the pipeline. We're at risk if we do not continue making those investments of having an interruption that would be deeply disturbing. The panelists today are going to discuss neurodevelopment disorders like cerebral palsy, autism, Down syndrome, which affect millions of people worldwide, and where research is critical and is promising. But that means the federal government needs to continue its investment. There's no substitute for the federal government providing the foundation for these research efforts. So I'm pleased to join with you to kick off this conversation today and urge everyone to be part of an effort to focus these critical elements for other people in Congress. We're always recruiting people to be part of the Neuroscience Caucus. We have regular briefings to try and enhance the understanding of neuroscience on Capitol Hill, particularly with the certified smart young people who really make Congress work. And so we're pleased that we're going to have an opportunity to have this program today and look forward to continuing this progress for the remainder of the Congress. Thank you very much.
Thank you so much, Congressman Blumenauer. Now let's move on to Congresswoman McMorris-Rodgers. She was first elected in 2004, and she represents the 5th District in the great state of Washington. She also is the ranking member of the powerful House Energy and Commerce Committee, and that oversees important health-related programs and initiatives. As co-chair of the CNC with Mr. Blumenauer, she's a longtime supporter of federal funding of the NIH. She's also a vocal champion for individuals with disabilities, and she's the co-chair of the Congressional Task Force on Down Syndrome. Congresswoman, please take it away.
All right. Good morning, everyone. It's great to be with you, and thank you for having me. And thank you for the incredible work that you're doing each and every day to advance brain research. I want to thank everyone at the American Brain Coalition for organizing this event, as well as my colleague and co-chair of the Neuroscience Caucus, Congressman Earl Blumenauer. He's actually my neighbor to the south. We both come from the Pacific Northwest, and we are proud to join together in co-chairing the Neuroscience Caucus again this Congress. As you know, the goal of our caucus is to promote better understanding of how the brain develops, functions, and ages. And our mission is to raise awareness and appreciate all of the work that you are doing to help us raise important awareness as to the research around the brain and about millions of Americans that are afflicted with neurological disorders of mental illnesses. The good news is that thanks to American ingenuity, we're on the verge of some major life-saving and life-changing breakthroughs. These breakthroughs would not only lift one of the biggest cost and care burdens on our healthcare system, it would transform the lives of millions of people. People like my son Cole, who has Down syndrome, that extra 21st chromosome. And the reality is that nearly 100% of people with Down syndrome develop the brain pathology for Alzheimer's. 1 in 3 seniors dies from Alzheimer's or another dementia. And deaths due to Alzheimer's have more than doubled over the last 20 years, increasing by 145%. As you all know, there is hope. America can win the future by leading a new era of research and development. And that's exactly what we are doing. I am excited to be the lead Republican on the Energy and Commerce Committee. And part of my goal and one of my top priorities is to advance legislation to empower more cures and medical breakthroughs, like 21st Century Cures that was signed into law in 2016. Thanks to cures, we're advancing life-saving treatments for very difficult diseases. And we should be doing more to build upon this law with innovative policies that encourage the rest of the world to follow our leadership for more affordable life-saving drugs and treatments. Our Neuroscience Caucus led again this year on the Brain Initiative, which aims at revolutionizing and understanding the human brain. By accelerating the development and the application of innovative technologies and researchers, we'll be able to revolutionize the dynamic picture of the brain. And this is just the start. I'm confident that with your help, Americans can win the future. And we can and we will develop new cures that will save lives and spare millions of people from these very difficult diseases. On behalf of families everywhere, I hope that you're encouraged today, and just know that you provide so much hope through your research and work. It's always great to be with you. Thank you.
Thanks so much, Representative McMorris-Rodgers and Blumenauer for being real champions for brain and neuroscience research. So on April 2nd, 2013, then President Obama announced the BRAIN Initiative. It's a mouthful. It stands for Brain Research through Advancing Innovative Neurotechnologies. The goal is to help researchers find new ways to treat and cure and prevent many diseases of the brain. It's a collaborative effort, a team effort among many government agencies and private sector partners. We in the ABC are proud to have recently become the newest member of the BRAIN Initiative Alliance, comprising federal and non-federal members and affiliates, the goal being to coordinate and facilitate communications from the members related to the BRAIN Initiative. We're going to hear all about that today. We have 4 brilliant speakers and maybe some time for questions at the end of the hour. But first, we're going to hear from Dr. John Nagai, and he's the director of the whole deal, the NIH BRAIN Initiative. He oversees the long-term strategies, the day-to-day operations for the initiative, and he's got plans for the next 5 years, a big plan that actually commenced in early 2020. So Dr. Nagai, I'll turn it over to you.
Thanks, Matt. If I can share screen, see if I still know how to do this. Okay, are you seeing my screen all right?
Yes.
Great, thank you. Thank you, Congressman Blumenauer and Congresswoman McMorris-Rodgers for your support, and of course to all of the members of your caucus for your support. We are both grateful for and inspired by all the trust you place in us to advance neuroscience, brain science in the interest both of discovery as well as for advancing human health. I just wanted to take a few minutes today to give you a 50,000-foot overview of what we're up to, a bit of an update, but really a look toward the future of where we hope to be or go in the next 5 to 10 years. So just to review for those of you who may not be fully up to speed on this, maybe very few of you in this room here today, the mission of the Brain Research through Advancing Innovative Neurotechnologies, or BRAIN Initiative, is to revolutionize our understanding of the human brain by accelerating the development and application of innovative technologies. One thing we're hampered by in terms of understanding how this incredibly complex organ works is we just don't have the tools to understand it. So this initiative was announced by then President Obama in 2013, with the first awards rolled out in 2014, and represents a partnership between 5 US federal agencies and private foundations. The efforts in the NIH BRAIN Initiative were guided by 2 really terrific strategic plans, the first from the BRAIN 2025 report issued in 2014, and then later a touch-up or revisit of the strategic plan from the BRAIN 2.0 reports in 2019. Now, the goal of the NIH BRAIN Initiative is to develop and apply new tools for understanding how neural circuits underlie complex behaviors, both in health and disease. In order to achieve these goals, it's very important for us to leverage emerging technologies to enable new discoveries about how neural circuits function, to use these discoveries as a foundation for therapeutic strategies for human brain disorders, and very importantly, to disseminate and democratize these technologies for basic discovery and clinical applications so that they can be used for the benefit of all. Now, I mentioned before the revisit of the strategic plan from the BRAIN 2.0 reports. These were commissioned by NIH Director Francis Collins. They were finalized and released in 2019. And there were 4 major take-home messages I'd like to just emphasize here from these 2 groups. One emphasizing the neuroscience itself, and the other the neuroethics aspects of what we do. The first was to stay on the productive path already underway. The group saw great success, and they encouraged us to continue along the same path. There was an urging to balance individual investigator research with team science. Now here, I would contend that necessity is the mother of invention. I think one of the great successes of NIH-funded research is this idea that the individual investigators are the source of creativity that come up with the great questions we need to answer. The goal of the BRAIN Initiative is to embrace those questions, to develop better technologies so we can ask and answer better and more precise questions. The third take-home message was to enhance the integration of neuroscience and neuroethics. In other words, to really be mindful of the broader societal implications of all that we do, not as an afterthought, but really to bake this into our research strategy from the very, very beginning. And then finally, They urge us to consider devoting ample resources toward standing up large-scale transformative projects based on the success of the BRAIN Initiative in the previous 5 years, as well as in the fields of biology and physics and engineering more generally. And here I just like to spend a little bit of time talking about the so-called BRAIN 2.0 transformative projects that we feel will transform the way we conduct neuroscience research and apply these lessons to treating human brain disorders for generations to come. So again, these were inspired by the BRAIN 2.0 Working Group report. And to put it in very somewhat simple terms, what we need is a parts list and a wiring diagram for the brain, this marvelous biological computer. If one wants to understand how a computer works, one needs to know what parts comprise that computer, how many do you have of each kind of part, what are the different properties of the different parts, and very importantly, how do they wire together? How do they communicate with each other? So we need a parts list and a wiring diagram for this marvelously complex computer. And we also need a way to interrogate and modulate these circuits. So by that, I mean, we need to test hypotheses about what these different parts do and how the different connections convey information and underlie behavior, and modulate them to test these hypotheses, but ultimately to modulate them in the cure or treatment of human brain disorders that involve brain circuit dysfunction. So let me just go through these with you very briefly. The first is the parts list project, the phase 3 of the Brain Cell Census Project, which builds on previous success of the Brain Initiative Cell Census Network, or BICCN, which started primarily focusing on a mouse brain census, but with also some attention to non-human primate and human brains. And the goal here is to shift the emphasis to humans to generate a comprehensive brain cell atlas for the human, and the target launch date for that is fiscal year 22, so next year. We will be standing up a connectivity analysis, a so-called wiring diagram project, to generate a connectivity map for an entire mammalian brain at multiple scales. We just finished off an intensive series of workshops co-sponsored with DOE Office of Science, and we intend to launch this project in FY23. And this just gives a very brief overview of some recent work conducted by the Brain Initiative Cell Census Network, highlighting the vast complexity of cell types in the mammalian brain, as well as the first glimpses of how these different cell types connect to each other. And this will be the basis of these transformative projects. And I should mention that the Cell Census Project has been accelerated in the launch through congressionally directed funds, so generously provided by Congress for FY21. And then finally, we need a way of accessing these cell types and circuits. So the goal here is to develop tools to access cell types in new and different ways in both rodent and NHP brains, as well as in human cells and tissues. This project was launched in FY21, again, accelerated by congressionally directed funds. The long-term goal here is to develop therapeutic strategies for human brain disorders. And Dr. Levy from the Allen Brain Institute will tell us a bit more about efforts in that domain. So I'll just pass that on to him. And I just want to finish with two brief examples of some amazing, I think, amazing developments in the Brain Initiative space, leveraging this synergy between biology, medicine, and engineering to develop treatments and cures for human brain disorders. So here is an example of a very recent paper published from Jamie Henderson and Krishna Shenoy's groups at Stanford University, where they developed a prosthetic device based on a brain-computer interface that allows a paralyzed patient to communicate with the outside world by thinking of letters that he wishes to write and converting that into a text application. So here, this is designed to restore the ability for people with spinal cord injuries or who might be locked in from ALS to communicate. And what they've done now is they've recorded from part of the cortex. They've interpreted the neural activity from the patient as the patient is thinking, as he's writing specific letters, and they converted that into text output. This patient can type at 90 characters per minute, which is comparable to the speed at which I can text on my iPhone with 95% accuracy, which is better than I can do on my iPhone. So this really speaks toward the amazing technologies and understanding of how brain circuits work, machine learning algorithms that can interpret this information, that can provide true relief to patients who are otherwise suffering from debilitating paralytic disorders. Now, this is all fun and well in the lab. What are we doing for patients in real life? Recent study from Dr. Phil Starr's group at UCSF has developed an adaptive brain stimulation device in Parkinson's disease patients based on wireless communication, where he has fully embedded electrodes in patients' brains. They can wirelessly stream data to a tablet device that allows unrestricted movements and allows the patient to move around in his or her natural environment at home. And what they can do is that they can identify signatures that can instruct the stimulator to intervene, but only when there's dyskinesia or improper movements going on. And this has the potential of reducing side effects of so-called open loop DBS stimulators that are always on. So this really is at the forefront of moving this new technology from lab to home. It relies on very sophisticated machine learning algorithms to read what's going on in the patient's brain and to intervene when the patient needs intervention to allow proper movement, and also relies upon new technologies and wireless communication, which is really the goal here is to move these technologies from the lab to the home. So I hope you can see that the discovery supported by BRAIN laid the foundation for novel interventions in human brain disorders. And we are now unraveling the circuits and computations underlying complex behaviors in identifying the cells and circuits affected in neurologic and neuropsychiatric disorders, which will lead to, as I just showed you very, very briefly, new sensory motor neural prostheses, closed loop deep brain stimulation devices, which can control the effects of circuit disorders in a much more fine-grained and precise way. Ultimately, we wish to move this research into targeting specific circuits affected in brain disorders using molecular and gene therapies. And this is the blue sky goal, but I think seeing the revolution in neuroscience and in engineering and computer science around us, I think this is an attainable goal in the next 5 to 10 years. And with that, I will end and just refer you to our website for additional information, and hopefully there'll be time for questions at the end of this session. Thank you.
Thanks, Dr. Nagai, for your exciting vision, your overview, your review of technology and teams and ethics and parts and wiring and systems and tangible advances to help people. This is really exciting. The whole is truly greater than the sum of the parts here. Next, we're going to hear from Dr. Diane Lipscomb. She's the director of the Robert J. and Nancy D. Carney Institute for Brain Science at Brown University. She's going to focus her talk on the National Science Foundation's NeuroNext Technology Hub. So please take it away.
Thanks so much. I'm just going to go ahead and share my screen. There we go. And someone's going to let me know if that doesn't look good. I think it looks good. Great. So, first, I really do want to add my thanks for the tremendous federal support that we've received for research, but also to stress that really basic research is the path to new discoveries and new cures and treatments, as so eloquently really highlighted by Representatives Kathy McMorris-Rodgers and Earl Blumenauer. And so I also want to give them a special thanks for their incredible support, and particularly being the voice for us, and really stressing how critical it is to continue this investment in research for multiple reasons that they laid out, and I'll come back to that at the end. We also heard, of course, that brain science diseases really impact everybody, myself included, family members, and it's so incredibly frustrating not to be able to do something to help, but research is really the way to new cures and treatments. And as pointed out, I'm going to just focus on the tremendous NeuroNext projects that have been funded through the National Science Foundation, consistent with the BRAIN initiative to develop new technologies, cures, and treatments, but did want to also highlight here at Brown the very exciting research that's also going on in the brain gate area. So as is shown from the first slide here, the bioluminescence is something that we've all seen and all interacted with. I was just down in DC and was surrounded by fireflies when I was sitting out on the porch at night. They're just so incredible, these tiny little insects emitting a massive amount of light. And so our project in the National Science Foundation funded team project is to think about ways to use bioluminescence, develop new technologies using this natural light source that is highly biocompatible, meaning, of course, that the organisms that are emitting this light using this natural source, that's not toxic to the organism. And so there's a lot of fantastic history here. These bioluminescing proteins have been used for many years and have captivated humans for a very long time. About 2,400 years ago, Aristotle noted that marine animals were giving off this cold light because bioluminescence doesn't give off heat, and that's one of its great properties. So I'm just going to give a couple of examples about what we're doing in this project, but also begin to talk also about the impact of that, the multidisciplinary team, and the training that goes on within this NeuroNext hub. So Dr. Nagai talked about the use of BrainGate and electrodes and electrical approaches using brain-computer interfaces to try to repair, also reroute or drive prosthetic devices. Extraordinarily exciting work. This technology that we're working on is a different approach, but to use this naturally occurring light from bioluminescing proteins harnessed from nature, optimized to be able to, for example, drive neural activity, dampen down neural activity, or increase neural activity in the nervous system. So what you're seeing here is this video where I'm showing a connection between two blobs here, and these are this is how the nervous system is communicating between one cell and another cell, and that communication is typically carried out by the release of molecules. And what I'm just showing you here is that we've been able to design releasing proteins from one neuron to another neuron to be able to form a connection, a bioluminescent connection, which could boost or actually replace the normal chemical connection. This could have, for example, applications in diseases such as amyotrophic lateral sclerosis or Lou Gehrig's disease, where there's a failure of communication between the motor nerve and the muscle. And so this is one example of where we could use this methodology, the natural bioluminescence, emitting light to drive optically activatable proteins, optogenetics, to repair or boost connections. And this has tremendous important applications in neurodegenerative diseases and potentially also in epilepsy. Here, I also want to stress the importance, we talked about individual and team-driven research, and I completely agree that both are really critically important. This is a team project in which we have a collaboration actually across the United States with Nathan Shaner, Nathan is in California, Uta Hochschwender, Central Michigan University, myself, and Chris Moore. And we each have particular levels of expertise, and we came together to form something that wouldn't have happened otherwise if we hadn't all come together as a team. And so I really want to just highlight the importance of bringing together experts and the incredible value of this NeuroNext hub type approach funded by NSF to pull together experts from across the country, across university and institute boundaries, to bring together to make this really highly effective team from molecules to animal models of testing the technology. The other thing important to highlight here is the kind of workshops that we put together. It was really highlighted that an important part of the funding that we get is to also disseminate and have really open science. So we make discoveries and we provide the tools and the technologies to the neuroscience community and to the brain science community in a very open and easy way. The other part of that, of course, is to train the next generation of young scientists who are going to be taking over from us. Very critical to make sure that there's a continuum of high-level research going on in this country, in the United States. And so these are undergraduates of the Marine Biology Lab in Woods Hole, Massachusetts, and they've got their nets and they were going out to capture new bioluminescing organisms from the waters around Woods Hole. In fact, they did, and every time they go out, they find something new, which is pretty exciting. So undergraduates from across the country. And then this slide, I'll show you kind of a little bit of a timeline capture. So all of these dots over the time course of our project, which is heading into its 5th year, final year. These are all the collaborators and the tools, showing a map across the United States of the tools we're developing, the students we're training, and the participants and the new collaborators that have been recruited into the project. So I would hit on 3 elements: the research, the novelty, the technology development. Within the research area, the importance of sharing open science and bringing and giving new tools to other investigators so that they can carry on and do the work that use the tools that we're developing. And in addition, training the next generation of scientists. And finally, just to reiterate things that have already been said and I think will be said, there is no shortcut to life-saving treatments. The urgency is now. And again, to underscore the very critical importance of the National Science Foundation and the National Institutes of Health to really lead in breakthrough discoveries for a better society, stronger economy, and also to train our next generation of scientists. Thank you.
Thank you, Dr. Lipscomb, for your illuminating talk on the NeuroNext Hub. Next, we're going to hear from Dr. Nicole Rust. She's an associate professor in the Department of Psychology at the University of Pennsylvania in the School of Arts and Sciences. And Dr. Rust, please take it away.
There we go. All right. So thank you, Dr. Rizzo. Let me also begin by thanking Representatives Blumenauer and McMorris-Rodgers for their leadership and their support of the Congressional Neuroscience Caucus. I also want to thank the American Brain Coalition for organizing this event and inviting me. It's a real pleasure to contribute. So I'm here as a researcher whose work is supported by a member of the Brain Initiative Alliance, the Simons Foundation. The work that we do is also supported by the National Institutes of Health and the National Science Foundation. And we work on memory. Today, I'll be telling you about the work we've been doing recently, and I'll be describing it as an arc from our basic investigations to how memory works in healthy brains, and then how we use what we've learned in order to develop new treatments for memory dysfunction, such as treating Alzheimer's disease. And I hope by the end of these 10 minutes, I will have convinced you, or at least elaborated on your understanding of why basic research is so important and foundational for developing these new treatments. So I'll just begin by digging into the basic research. So the type of memory that we study, it's called visual memory. It refers to our ability to see things and remember what we've seen. And what motivates us to study this is that we as humans are just really remarkable at remembering what we've seen. So the way we study this in the lab is we have participants come in and look at very large stacks of photographs, so thousands and thousands of images. And we have them flip through these images, looking at each only once, only for a few seconds. Flipping through these images will take them many hours. And after they've seen all of these photographs on single exposures, we'll give them a memory test where we place 2 images side by side, one they've seen and one that they haven't, and we'll ask them, which one did you see? And systematically, people perform really remarkably well at this memory test. They're often over 90% correct. And this is true despite the fact that the content of the images can be very similar, like these 2 images here. So we've known about this remarkable form of visual memory for quite a long time now. And what we don't understand is how our brains manage to support it. So how is it that our brains manage to support thousands of memories after seeing pictures only once and only for a few seconds? This is what we don't understand. And so this is what the basic research in my lab is focused on trying to figure out. So in the past handful of years, we have been focused on identifying what we call the brain's memory signal as a first step toward understanding memory. And just to explain to you what that means, I'll remind you of an experience I imagine you've all had, where you're walking down the street or the hall and you see someone and you immediately know that you know them. But often for a few moments at least, you can't recall anything else. So you can't remember who they are or how you know them. All you know is that they're familiar to you. When you have that experience, there must be a signal somewhere in your brain that's activating it. And the question is, where is that signal located in your brain? And how is it reflected in brain activity? That's what we call the memory signal. Now, in order to figure this out, it turns out we can't investigate the brains of humans because answering this question requires recording brain activity at the resolution of individual nerve cells, neurons. And the techniques that are available to us to do this in humans, the non-invasive techniques, simply don't have that resolution. At best, they reflect the combined responses of about 10,000 neurons that are firing at the same time. So in order to understand the brain's memory signal, we investigate animal models where we can both record neural brain activity at that resolution, and we can also investigate new treatments for memory dysfunction, as I'll be describing in a moment. In my lab, the animal model we work with is the rhesus macaque monkey. And this is because rhesus monkeys are the only animal that has been demonstrated to have visual memory that reflects our own. So the only animal that can look at large numbers of images, see each only once, each for a few seconds, and then accurately report whether or not they've seen them before. No other animal has been demonstrated to do that. So in rhesus monkeys, as they're performing the same memory tasks that are used to study memory in humans, we record brain activity at the single neuron resolution. And we do that using the same sorts of approaches that are actually used in humans. In this case, in patients that are being diagnosed for intractable epilepsy. So we use those same techniques to record neural signals in their brain. And then the last step involves interpreting that brain activity, which tends to be quite complex and very difficult to understand. And so this is why we work with quantitative types that are such a central and important component of the brain initiative. So here I'm referring to the physicists and the mathematicians, the engineers and the computer scientists that help us develop new ways to analyze all of this big data and use it to interpret what's going on in the brain. In our case, we often work with a director at the Simons Foundation's Flatiron Institute. His name is Eros Simoncelli. He's also a professor at New York University. So these are the approaches we use to try to figure out how the brain works and how this memory signal is reflected in the brain. And we've applied those approaches to successfully identify what we think is a really good candidate for the brain's memory signal. It turns out that this memory signal is reflected in the same parts of our brains that are responsible for helping us see as well as remember. And so I'll just describe how this works. So when you're looking at a particular thing, like let's say you're looking at a pencil, this will activate your eyes to activate in turn your brain. And your brain's job is to figure out what is out there in the world, what are the objects out there in the world in terms of interpreting that from the brain, the activity that's going on in the eye. Now, once it's figured this out, the way it signals to the rest of the brain, the content of what's going on out there in the world is through patterns of activation of these individual neurons that I was talking about. So you get one pattern of activation for, say, a pencil, you'll get a different pattern of activation for a coffee cup, and so on. This is how your brain interprets what it is that you're looking at. In addition to this activation that tells you what you're looking at, this activation also tells you whether or not you've seen it before. But in terms of signaling memory, this information is signaled by the vigor of the overall response. So you get more vigorous responses the first time you see something and less vigorous responses when it's familiar to you. And this is the memory signal in the brain. So it's a reduction in the overall response that triggers you to remember that you've seen something before. This description of the memory signal has been held up to be robust across many different experiments and many different challenges. So now that we've identified a good candidate for the brain's memory signal, we're now building on that understanding to try to figure out how to improve memory and treat memory dysfunction by asking what can make this memory signal stronger. And one of the avenues we're pursuing is looking into the interaction between memory and alertness. So we know it's the case that if you're well rested or if something surprises you, you'll remember it better than if you're sleepy or if you're bored. This suggests that memory and alertness interact in some way in the brain, and we're working to figure out how we can leverage that interaction in order to treat memory. So this is work that I am doing in collaboration with an investigator at my institution, Dr. Joshua Gold. Dr. Gold is an expert in alertness and how it's mediated in the brain by a chemical called norepinephrine, which is released by a structure called the locus coeruleus. And the way we're looking to see how memory and alertness interact with one another is to do basic research on the interaction between norepinephrine and its release in order to enhance memory or modulate memory function. So that's a basic component of research in our lab that we're collaborating on. In addition to this basic understanding of how memory changes with alertness, we're also working on a new treatment for memory dysfunction, which builds on the knowledge that this brain area, the locus coeruleus, which releases this chemical, is one of the first stages, the first parts of the brain that's impacted in early stages of Alzheimer's disease. As we all know, Alzheimer's is a completely debilitating disorder, and we currently don't have any good treatments or cures for Alzheimer's. So we're working on the idea that if we can stimulate the locus coeruleus to release the appropriate amount of norepinephrine at the appropriate time, meaning when it is that Alzheimer's patients need to remember, this might be a treatment that can mitigate some of the detrimental impacts in memory in these patients. And the way we're working to do this is through stimulation of the vagus nerve. So the vagus nerve is a nerve that is accessible on the neck, and this forms a minimally invasive type of treatment because it can be accessed via simple surgical implantation of a cuff here. Vagus nerve stimulation is currently being used clinically to treat intractable epilepsy as well as depression. And we have good reason to think that stimulation of the vagus nerve stimulates the locus coeruleus in order to release epinephrine here. So this idea is what we're building on in order to try to develop this new treatment. The big challenge in developing this treatment is that there are many, many different parameters associated with vagus nerve stimulation. And exploration of all of these parameters is currently intractable and unsafe in human patients. So what Dr. Gold and I are doing is we are leveraging our understanding of how memory and alertness interact in the brain, coupled with our animal models in order to figure out the parameters that may be effective at using vagus nerve stimulation to treat Alzheimer's disease. And while we're still at these very early stages, what I hope is that this description of how basic research can inform a new treatment for memory gives you some foundation, some context in order for understanding how this process works. So with that, I will just end. Thank you. And again, thank you to our sponsors, for which we are so very grateful.
Thank you so much, Dr. Rust, for that great talk on memory and how to make it better. We're going to move on now to hear from Dr. Boaz Levi, or Levi. He's the Associate Investigator at the Allen Institute for Brain Science. And please take it away.
I'd like to thank the organizers for inviting me to present our work from the Allen Institute. I'd also like to thank the congressional support of the Brain Initiative that we've been the beneficiaries of. My name is Boaz Levi. I'm an associate investigator at the Allen Institute for Brain Science, and today I'm going to tell you how we go from brain mapping to gene therapy. So John and I did an excellent introduction, but the Allen Institute, as well as other people, are really focused on understanding the brain from a bottom-up approach. If you're confronted with a really complex object, such as an automobile, a sensible approach to understanding how it operates is to take a reductionist approach and to explain and describe all the various parts that go to building a functional vehicle. We take the same reductionist approach in brains, where we like to describe each of the individual brain cell types so we can better understand how these parts are built and how they work in concert. We can describe these cells based on their shape and their morphology, by their electrical firing properties, and this is how these individual neurons communicate with each other, And really critically, we like to characterize the molecular properties of individual cells, largely based on the genes that they express, and we can measure these and show how one cell type is different from another cell type. The brain-supported studies that we've undertaken have revealed gene expression and regulatory landscapes of mammalian brain cells. We tend to focus our work on mouse, which is the most commonly used model organism for neuroscience, as well as human, because we want to be able to translate all of our discoveries to improve human health. We tend to take single cell resolution approaches. We look at gene expression of individual cells. This is how the genome encodes instructions for the cells to become what they eventually become. And by measuring the gene expression of many different cell types for a given brain region, we can describe this beautiful taxonomy of cell types. Any given region of the brain, this is from the motor cortex, has over 100 different cell types that are present, and they can all be distinguished from each other based on the genes that they express. In addition, we also characterize brain regions based on their epigenetic profiles. Epigenetics reveals pieces of the genome that control the differential expression of specific genes in the brain. And by doing this kind of analysis, we can reveal these small pieces of the genome that control gene expression. And what's interesting is many of these regions are enhancers, and they are selectively used in cell populations, such as this enhancer is only used in this particular population of neurons and not these other ones. So these molecular characterization studies of brain cells can be leveraged to build precision viral genetic tools. And so I'm going to talk about viruses, and these are not viruses like COVID-19, which causes disease. We use adeno-associated viruses. These are commonly found in the human population and are not known to cause any diseases. However, they're an excellent delivery mechanism to transmit genes into all sorts of species. We focus on characterization of the epigenetic profiles, which reveal selective packets of the genome to control gene expression. What's really exciting is we can take these regions, these enhancer regions, and put them in a viral vector to drive the expression of a fluorescent reporter, such as yellow fluorescent protein. These can be packaged into viruses and injected into a mouse brain to see if we can recreate the expression pattern that was predicted from the epigenetic studies. And what we can see is when we inject these into a mouse brain, and these are images of mouse brains, we can label neurons as well as non-neuronal populations. We can label all neurons, and here where you see white is where our fluorescent tracer is being expressed. We can also label subsets of neurons, and here we can hone in on specific circuits in the brain. We can label these non-neuronal populations as well using these viruses, including astrocytes and oligodendrocytes, and these are really critical to the functioning of neural circuits and are often seen to go awry in disease states. Since we're committed to translating our technologies and our viruses to improve human health, we want to show that our virus tools actually work across species. So here are 5 viruses. Each virus has a different enhancer element to drive gene expression, and we can see this virus expresses in astrocytes, another in oligodendrocytes, and several different types of neurons that are easily distinguished here just based on their shape and position in the brain. We've injected these same viruses in macaque monkeys, which are a close model of humans. And what we can see is excellent conservation of specificity of many of these viruses. We can see this by shape alone, but also more detailed characterization studies show that these are actually the same cell types, but in different organisms. And this really gives us confidence that the tools that we're building and validating first in mouse actually may be able to behave in a predictable fashion when injected into humans, making them an excellent template for a gene therapy. Importantly, we have new collections of viral genetic tools that we've built that target discrete cell types and circuits that are really key in disease. For instance, there's a very important structure called the basic ganglion or ganglia, and this controls the information flow from the periphery up through multiple brain structures into the cortex where the higher order processing happens, and back down through the striatum and other brain regions through the spinal cord. Basoganglial dysfunction has been shown to be present in a variety of neurological disorders, including Parkinson's disease, dystonia, Huntington's disease, Tourette syndrome, addiction, autism spectrum disorder, obsessive compulsive disorder, as well as a number of other diseases. And what's really exciting is we are beginning to build these highly specific and penetrant viral tools that can target gene expression to these structures. The brain is shown by the blue stain here, but our virus, although it's present in many of these cells throughout the whole brain, is only driving gene expression in these green cells. And here we can target these very important medium spiny neurons that have axons that project to other targets. In addition to the basic ganglial structures, we can target other cell types that we know are defective in many disease states, such as Purkinje cells, which are defective in ataxias, astrocytes, which are implicated in Alzheimer's disease, as well as oligodendrocytes, which are defective in Krabbe or other demyelinating diseases. So these viral genetic tools constitute a direct path from brain-supported basic discovery work to therapeutic intervention. So we start with our epigenetic profiling, which has been really well supported from the Brain Initiative funds. We can identify these very selective regions of the genome where we can drive selective expression. We can put it in a virus and drive our reporter expression very selectively through the brain in multiple species. These are really valuable research tools for basic neuroscience. And because of this, we want to make them freely available to anybody. And so we release these viral tools through Addgene, where the rest of the research community could access them. However, some of our vectors target brain structures that are really key in disease and disease misfunction. We can take a very similar strategy where we can use that same vector that has the enhancer for specificity, and instead of driving a reporter, we can drive a therapeutic transgene and put it in an animal and see if we can rescue a disease phenotype. And in this case, we're targeting a genetic epilepsy, and we can show that a specific therapeutic virus can rescue this phenotype. We consider this as a really exciting advance, and we think that these viral tools are going to be very valuable for targeting many different genetic neurological disorders, and we're really excited to move forward to translating our basic research tools to therapeutic interventions. And so with that, I'd like to thank everybody for listening to I'd like to thank the Allen Institute. We take an open science and team science approach. This is a big institute in Seattle, Washington, and these projects really require a lot of help from many experts. I'd also like to call out the drivers of the Viral Genetic Tools Project, Jonathan Ting, John Mish, Basilica Tasic, and Tanya Daigle, as well as our fantastic leadership from Hanghui Zhang, Ed Lean, and Christoph Koch. And last, we've been the lucky recipients of really fantastic support from the Brain Initiative. There's a number of different grants that have supported different aspects of this work. So thank you for your attention.
Thanks very much, Dr. Levy. I know people have lots of burning questions, but we're about at the end of our hour, and you can feel free to ask those offline, and we'll be glad to answer them. But I want to say that what a great time to be alive, to hear about these amazing advances to help diseases that have no cures, that have had no treatments, to hear from Dr. Lipscomb about making connections with bioluminescence to maybe cure Lou Gehrig's disease, to hear from Dr. Rust about mechanisms of memory and multidisciplinary approaches to improvement, including with neurosurgical techniques and VNS. Dr. Levy for the many different pathways that you suggested for sort of looking at the cell zoo of the brain and making connections and all sort of therapeutic interconnections or interventions with genetics. But we have come to the end of the hour. I want to say that I'm really grateful to members of Congress and their staffers, to the ABC members, and other supporters of neuroscience research and the Brain Initiative for being on the Zoom today. We have a couple of requests for members of Congress and staffers. And the first is to assure robust and sustainable funding increases for biomedical research, including the amazing Brain Initiative research that you heard about today. A second request is that for the House staff, if your boss isn't a member of the Congressional Neuroscience Caucus, please suggest that they join. Again, I want to thank the speakers for their inspirational insights today. And thanks to the Congressional Neuroscience Caucus for hosting today's briefing. Again, a special thanks to Congresswoman Kathy McMorris Rodgers and to Congressman Earl Blumenauer for their opening remarks and for their enduring support. I want to thank you all for joining us today. Have a great summer and see you next time. Bye-bye.